Hello, class. I hope that you have looked at the anatomy for the nervous system, and at the end of that, you saw that I showed a few slides of some notes. Here, we're going to go through the notes specifically for nervous. The other showed a little bit of nervous and special senses, and then next week, we'll do the anatomy and the notes for special senses. So I'm hoping that you are taking the time to look at this stuff now so that you can get the special senses under your belt and have time to ask me questions about anything that you need clarity on before the exam. So the notes are posted. You want to make sure you are going through the notes and viewing the lectures at the same time and writing down anything that you need to add to the notes. And also make a note of anything that you need to ask me questions about, so that you can get clarity over anything that you need to have clarity over. So I'm going to turn me off, and we're going to get into the notes for the nervous system. So the nervous system is a rapid acting regulatory system, right? So this allows us to take in information, to perceive some kind of stimulus, and then respond to it. And so in the most simple way to look at this, we have sensory input, we integrate and analyze that information, and then we have motor output. Whether or not it's a reflex or whether or not it's something that requires higher level thinking, it works the same way. We have some input, something that's giving our nervous system some information. Our nervous system decides what we should do with that information and generate an output. So if we look at the top here, the sensory input, we have an eye. So maybe we see a stop sign. That information comes in, and we have to think about, what do we do with a stop sign? Now, sometimes we think about it so quickly because it's something we have become accustomed to, then we respond right away. That's not exactly a reflex. We still have to think about stopping at that stop sign. And so then we have motor output. Put your foot on the brake, right? Here, sensory input is, I want to drink this glass of water. I may be thirsty. I have that input to drink, and then I drink and by picking up the glass. And so I tell myself, okay, pick up this cup, drink the water. Okay. So sometimes this can be involuntary, which is a reflex, which we'll talk about later. So we have some sensory input. We monitor some kind of change in the environment. Could be an internal change. And then we have integration. What does this incoming information mean? What do I need to do about it? Then we generate an output. So then we have 3, which is a motor output. After we have analyzed what that input meant, we need to do something about it. And this is in the simplest of terms. We've talked about the neuron before. We talked about this when we talked about anatomy, so I won't spend a whole lot of time on this, but you got the cell body, or the soma, the axon hillock that connects the cell body to the axon. The axon is wrapped with Schwann cells. The Schwann cells secrete myelin, which is an insulator. And then between the Schwann cells are the nodes of Ranvier. And the impulse is going to jump from node to node, and we'll talk about that later. And then you have the axon terminals and the terminal buttons. Okay. And so the neuron is the tissue, the cell that creates the tissue for passing impulses. Neurons do not continue to carry out mitotic division once they're mature. So if somebody has a neurological tumor, right, a brain tumor on their spinal cord, it's going to be from some other type of cell, not a neuron, because they lose their ability to divide. And tumors are essentially mitosis gone haywire. And then you get too many cells and you get a mass. Different types of neuron based on shape or morphology. So multipolar is the one we typically look at, the multipolar neuron. Then you can have unipolar neurons and bipolar neurons. And we have one also on this picture called anaxonic neuron. So let's zoom in on this picture and look at the differences here. So our multipolar neuron is the one we typically talk about. And this one, we have our cell body and our axon, and we got the dendrites leading to the cell body, and then the axon terminals leading away from the axon. In a unipolar, we have a cell kind of sitting off to the side, and then we just really have one extension. That one extension has a dendritic end and an axonal end. But if you look at it, that's all continuous, right? So the cell sits over to the side, kind of like a T, right? And the axon and dendritic area is all connected together. Okay. And then we have a bipolar neuron. We got a dendritic extension and an axonal extension coming from different sides of our cell. And then an anaxonic neuron. This is a neuron that has a lot of dendritic ends, but not a specific axon. Okay. Couple of definitions. Nerve cell bodies can be clustered within or without the central nervous system. So two parts of the nervous system, the central nervous system and the peripheral nervous system. The central nervous system is the brain and the spinal cord. The peripheral nervous system is all of the nerves that extend from the brain and the spinal cord. And so when we have nerve cell bodies within the central nervous system, they're called nuclei, or one is a nucleus, and then ganglia or ganglion is one in the peripheral nervous system. The nerve fibers are bundled together to create either nerves or tracts. So we use the term nerves in the peripheral nervous system or tracts in the central nervous system. And then white matter and gray matter. White matter is myelinated, gray matter is unmyelinated. Let's zoom in on these pictures. So ganglion is where we have that cluster, right, at the peripheral nervous system. A nucleus within, this is in the spinal cord, and then tract in the central nervous system. So we have tracts inside of the brain here, but then outside we have nerves. So it's a tract in the brain, and then once we extend outside of the brain, we see a nerve, and then here we see nerves. And gray matter and white matter. Gray matter, this is in the cerebrum, is more superficially located, and then white matter is deeper. Okay. I want to just flip back for a second because I want to talk about the central and peripheral nervous systems. Even though you saw these notes, I didn't talk about them. So I'm going to flip back and forth a little bit. So the two divisions of the nervous system I just alluded to, the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, all of the nerves that extend from the brain and the spinal cord. And so when we talk about the peripheral nervous system, we have two divisions, sensory and motor. And so we've used those terms already, sensory and motor. Sensory is the afferent division. Afferent or afferent means incoming, sending impulses to the central nervous system. The motor is the efferent. Efferent or efferent means outgoing, sending impulses away from the nervous system to the effector organs. Okay. And then the motor division is further divided into somatic and autonomic. Somatic and autonomic. So somatic is voluntary, where we can control our skeletal muscles, and autonomic is involuntary, and that includes all of the glands, the cardiac muscle, and smooth muscle. And then autonomic is further divided into sympathetic and parasympathetic. Okay, let's see. So this is what I want to look at. So we have our central nervous system, which is the brain and the spinal cord. Then we have our peripheral nervous system, all the nerves that extend from the brain and the spinal cord. It has two divisions, sensory and motor. Okay. And I don't even talk about differentiating the types of sensory. So we just got sensory and motor. And then the motor is further divided into somatic and autonomic. And somatic is voluntary. Autonomic is involuntary, and that's further divided into sympathetic and parasympathetic. So I want you to have an idea of this flow chart. Let me do some things here so that you can write this down. I'm going to get rid of information that I'm not even requiring you to record. Okay, so I want you to put this flow chart down. So we have our central nervous system and our peripheral nervous system. Our peripheral nervous system is divided into two, sensory and motor. Then motor is divided into two, somatic and autonomic. Then autonomic is divided into two, sympathetic and parasympathetic. So write down this flow chart. Now, I do talk about this later in the other set of notes, but since I mentioned it here, I wanted you to get it down here. So we'll go back to the other set and continue. Okay. So we said that if you had some kind of neurological tumor, that what would be responsible would not be neurons, but other cells within the nervous system. So there are a number of other cells in the nervous system, either called neuroglial cells or glial cells. And these cells, in some way, support neurons, but they don't carry impulses. And so we have a number of them, and they function differently. So we have astrocytes. Those are in the central nervous system. And what they do is they wrap around the capillaries and help form what's referred to as the blood-brain barrier. And they also help keep the neurons in place. So they anchor to the neurons and they surround the capillaries. And then we have microglial cells in the central nervous system, and they engulf foreign material and dead cells. So they're phagocytic in nature. And then we have ependymal cells, and you may hear people pronounce them ependymal cells. They line the fluid-filled spaces in the brain and the spinal cord. So they line the ventricles and that central canal of the spinal cord. And then we have oligodendrocytes, and oligodendrocytes secrete myelin or form myelin in the central nervous system. And then, we have Schwann cells. They're also called neurolymphcytes, and they're in the peripheral nervous system, and they secrete myelin. And then we have satellite cells that protect and cushion nerve cell bodies for those unipolar neurons. Okay, so let's look at these pictures up close. So here's my astrocyte, and you can see that it has all these extensions extending from it. Oh, wrong pen. And you see all these extensions extending from it, and we'll see them, I think, in place down further on the other slide. And then we have microglial cells that will engulf foreign material and dead cells. Ependymal cells. Ependymal cells almost look like cuboidal epithelium that's ciliated. It's not, but it can put you in mind of that. The oligodendrocytes have the extensions that wrap around nerve cells and secrete myelin in the central nervous system. And then here are Schwann cells that wrap around and secrete myelin in the peripheral nervous system. And then here are those satellite cells. So they are surrounding the cell body of a unipolar neuron. And let's look at the picture below. So here's my astrocyte, and you can see the extensions wrapping around the capillary and anchoring to the nerve cell. So you see that here and here. Here are some microglial cells that are floating around, engulfing things. Here are my ependymal cells lining a ventricle. Oligodendrocytes wrapping around cells secreting myelin within the central nervous system. Anytime you need to make the screen bigger, hit that 4 arrows out in the right-hand bottom corner of your screen, and that'll make your screen bigger. So when we talk about sensory or afferent neurons, or the neurons that send information in, sometimes you have specialized dendritic ends or receptors. So we looked at some of these in the skin already. So we had naked nerve endings in the skin. We had Meissner's corpuscles and Pacinian corpuscles in the skin. Meissner's for light touch, Pacinian for deep touch. And then in the muscle, we had muscle spindle and Golgi tendon. So we've seen some of these receptors already, right? And we'll talk about more receptors when we get to special senses. So the muscle spindle and Golgi tendon are proprioceptors. They have to do with you being able to detect movement and stretch and tension and that kind of stuff as you're moving your muscles. Okay, so we have here our Pacinian corpuscles, our Meissner corpuscles, naked or free nerve endings. We didn't talk about Merkel discs, so we don't have to. Let's zoom in on these pictures. So again, you see our Meissner's corpuscles for light touch, Pacinian corpuscles for deep touch, naked nerve endings or free nerve endings often associated with pain. And then we have within our muscle spindle and Golgi tendon, and they have to do with tension and stretch and that kind of thing. Okay. Okay, so when we talk about how the nervous system functions, it actually functions electrically. And this electricity comes from the placement of ions, particularly sodium and potassium. And so all cells, particularly nerve cells and muscle cells, have what's referred to as a resting membrane potential. Okay. All cells have this resting membrane potential, but it becomes very important for excitable cells like nerve cells and muscle cells. So the resting membrane potential is established because the outside is more positive relative to the inside. The outside has a higher concentration of sodium, and the inside has a higher concentration of potassium. And that creates this resting membrane potential in the nerve at about negative 70 millivolts. And so when we have flow of electrical current from one point to another, that's going to be the current. Resistance is the hindrance of any flow. And so you just need to be familiar with Ohm's law, which is the current equals the voltage divided by resistance. And we're going to talk about this depolarization and repolarization in a bit. Okay. So how is an impulse passed down the nerve? And we kind of talked about muscle contraction, but we did not talk about too much of what was happening with the sodium and potassium. Well, we're going to talk about it here, and it works pretty much the same way with the muscle. So we start off with the resting membrane potential established, and the cell is said to be polarized. And when something is polarized, that means one end is the opposite of the other end. Well, in this case, our ends are the inside and the outside. Okay. And so the outside of the cell has a more positive charge and a higher concentration of sodium. And the inside of the cell has a more negative charge and has a higher concentration of potassium. So that establishes my resting membrane potential. So the cell at that point is at rest and is polarized. Okay. And then we get a stimulation. And that stimulation causes sodium to move into the cell. When sodium moves into the cell, now the inside of the cell becomes more positively charged. This is called depolarization. So when sodium moves in, that changes the resting membrane potential, and this is depolarization. Now the inside of the cell becomes more positively charged. That impulse is passed down the length of the neuron. That's called propagation. Once the impulse has passed, then the cell needs to go back to its original charge, or it needs to repolarize. So to repolarize, what happens is the potassium goes out. And when the potassium goes out, now the inside becomes negatively charged again, and the outside becomes positively charged again. That is called repolarization. So then we repolarize to make our charge positive on the outside again and negative on the inside again. But our ions are out of place. So our last step would be having the sodium potassium pump move the sodium back outside 3 at a time and the potassium back inside 2 at a time. Now, similar to when we talked about muscle contraction, I'm not concerned about you memorizing this in 4 steps, 5 steps, 12 steps. I'm more concerned about you understanding this process. Okay. Now, I didn't mention hyperpolarization. You can write down that definition, but I want to show you how this works. And by the way, this whole process, depolarization followed by repolarization, is called an action potential. So we start off at rest. Let's zoom in on this picture. We start off at rest. The cell is polarized. The outside is positive relative to the inside. Now, this is relative. So we got sodium at a higher concentration outside and potassium at a higher concentration inside. Okay. Then we get a stimulation. And our stimulation starts sodium to moving in. When sodium moves in, now that's making the inside become more positively charged. That continues to pass down the length of the neuron. The passing down the length of the neuron of this stimulation is called propagation. Okay, let me put that term here because they don't have it here. propagation of our action potential. Okay, so now the sodium moved in, we got depolarization. Now that's going to happen down the length of the neuron. So we have to get enough sodium coming in to meet a threshold. And the threshold is then going to ensure we're going to have a full stimulation followed by recovery, or full depolarization followed by repolarization. That whole process is an action potential. Then once the impulse passes the area, now we're going to start to repolarize. So then you see potassium moving out where we are repolarizing. Okay, and that gets moved down the length of the neuron, and then the whole thing will go back to its normal charge. But I'm going to add a number 6 here. The number 6 is the sodium potassium pump. Why? Because we moved sodium in and potassium out. We got our charge back to normal, but our ions are out of place. So I'm going to add a number 6. And I'm going to be consistent with the color that they were using. Number 6 would be moving the sodium back out and the potassium back in. We move 3 sodiums for every 2 potassium by way of the sodium potassium pump. And then that allows us to go back to our normal and do it all over again. So I say that at the end here. Okay, so we started with sodium at a higher concentration on the outside, potassium at a higher concentration on the inside. The outside has a positive charge relative to the inside. The inside has a negative charge relative to the outside. This cell is polarized. It's at rest. The resting membrane is a potential is established. Then we get a stimulation. Sodium starts to trickle in. I got to get enough sodium in to reach a threshold. Once I get that amount of sodium in, now I'm going to go into depolarization. So the whole length of this neuron, sodium is going to rush in and depolarize this neuron. That's going to be spread down the length of the neuron. The spread is called propagation. The whole process of stimulation or depolarization followed by recovery or repolarization is an action potential. As this is being propagated, the earlier parts start to recover or repolarize. That repolarization happens as potassium leaves the cell. So the potassium leaves the cell, and then once that's fully done, now our charge is back to normal, positive on the outside and negative on the inside. However, our ions are out of place because we started with sodium outside and potassium inside. So we have to put them back in place. So our last step is the sodium potassium pump, moving sodium out 3 at a time for every potassium every 2 potassium it brings back in. Okay. If you have any questions about that, please ask me. So we mentioned the fact that the impulse kind of hops between our neurons here, between the nodes of Ranvier, rather, here and here and here. So if I don't have myelinated neurons, are much more rapid acting because you can stimulate a depolarization at each of these nodes. Whereas an unmyelinated neuron, I gotta go step by step by step by step by step this whole way in one direction. And so it takes a whole lot longer. But when we have a myelinated neuron and I can trigger at each node, then my depolarization can move along much faster. Okay, so myelinated neurons are more rapid acting. Types of nerve fibers based on the myelination and the rate of conduction. So those that are more heavily myelinated are faster. Those are group A, so they're larger in diameter, more heavy myelination, and we see those associated with certain sensory and motor fibers. And then group B have a moderate level of myelination. Okay, and then group C are unmyelinated, and they are the smallest and the slowest. We talked about synapses before. A synapse is a point of connection or a junction between neurons, or a neuron and a muscle, a neuron and a gland. So it's where the terminal button of a neuron touches something else. Right? It can be touching another neuron, it can be touching a muscle, it can be touching a gland. That's a synapse. Let's zoom in on this picture. So here I want to talk about, this is one neuron, and this is the terminal button of that neuron touching the cell body of this next neuron. That is the synapse. So then they blow up the synapse here, and I'm not really sure why they show it this way, because this is touching a cell body. So this receiving end should actually look like a cell body and not another terminal button. Okay, so this would be the cell body here that is touching. And I'm actually just going to fill that in for the sake of it, because that's really what it should look like. This should all be filled in as the cell body. So all of this is just cell body. It's not another dendritic end. Right? And so on this cell body, you have some receptors for the neurotransmitters that are released from the vesicles of this terminal button. Okay, and so these are the receptors here for those neurotransmitters. So when a nerve synapses with something, there are neurotransmitters there. So again, this is at the cell body. I'm not sure why they made it look like it was with the dendrite or another axon terminal. But whatever it touches, that thing that it touches is going to have receptors for the neurotransmitter. So we can have electrical synapses. These are seen at gap junctions. We talked about gap junctions with smooth muscle and cardiac muscle, and they allow for electrical signaling or ions to pass from one cell to the next. And then we have chemical synapses, and that's what we're talking about here, where we actually are releasing neurotransmitter, and then the other receiving cell or tissue has receptors for those neurotransmitters. The space between, let's zoom back in, that tiny space in between here, that's called a synaptic cleft. So the neurotransmitter is released into this space, which is called the synaptic cleft. Okay. So we can have different types of synapses, and I don't think this is in your notes, so you want to make sure you write this down. So we can have different types of synapses. So we can have an axiodendritic synapse, where we have an axonal end or terminal button touching the dendrite of the next neuron. We can have an axiosomatic synapse. That's what we saw before, where the axonal end or terminal button is touching the soma. We can have an axioaxonic synapse, where the axon is, the terminal button is touching the axon. Okay. So axiodendritic, axiosomatic, and axioaxonic synapses. Axiodendritic, the terminal button is touching the dendrite. Axiosomatic, the terminal button is touching the cell body or the soma. And then axioaxonic, the terminal button is touching the axon. A couple definitions. A synapse, with regards to what the neurotransmitter can cause to happen, can be an excitatory postsynaptic potential or an inhibitory postsynaptic potential. So in excitatory postsynaptic potential, we get this rapid change that's going to lead to excitation. And then in inhibitory postsynaptic potential, we get this change that is going to reduce the ability to cause an action potential or inhibit stimulation. Okay, so we have here is our, let's zoom in on the picture here. Our resting membrane potential, and then we have some excitation, right? And we see the change here associated with that. We get some depolarization. Inhibitory postsynaptic potential, this is our resting membrane potential. And so we inhibit that change, and we might get some hyperpolarization or decreased potential for there to be any action potential or depolarization happening. So actually hyper polarization, even dropping below our resting membrane potential. Okay, so this is what we were talking about before when we briefly looked at the other page from the other slides. The divisions of the nervous system. So we have the central nervous system, which is the brain and the spinal cord. We have the peripheral nervous system, which is all of the nerves that extend from the brain and the spinal cord. The peripheral nervous system is further divided into sensory and motor. Sensory is afferent or afferent, meaning incoming. So then the sensory is going to send information into the central nervous system. And then we have the motor, which is also called efferent or efferent, which is outgoing. And so it's going to send information out from the central nervous system. Then the motor can be further divided into somatic and autonomic. Somatic is voluntary. We have control over that, right? Control over our skeletal muscle, for example. And then autonomic, we don't have control over. And autonomic is going to involve stimulating smooth muscle, cardiac muscle, and glands. And that's further divided into sympathetic and parasympathetic systems. Okay. Now, we want to differentiate between sympathetic and parasympathetic systems. But before I do that, let me just check and see. I want to make sure you have this down. We looked at this before. That's what I had you write down from the other slides. And let's make sure you do that. Okay, now let's go back and talk about sympathetic versus parasympathetic systems. So the first thing I want to put here is that sympathetic system is often referred to as the fight or flight system. So let's put that underneath here. And the parasympathetic system is often referred to as the rest and digest system. Okay. So now both of these systems have two neurons in place. One is a presynaptic neuron or preganglionic neuron, and a postganglionic neuron is the other. Preganglionic or postganglionic fibers. So in the sympathetic system, this is the system at levels of high stress, fight or flight. So if you have to fight or flee, what's going to happen to your blood pressure? It's going to go up. What's going to happen to your heart rate? It's going to go up. What's going to happen to your breathing? It's going to go up, right? If you are resting and digesting, all of those things go down. You're more relaxed, okay? So for the sympathetic system, the neurons originate from the thoracic and lumbar nerves, thoracic and lumbar regions of the spinal cord. And the first neuron is fairly short. I wouldn't say it's super short, but fairly short. And it releases acetylcholine. And then the second fiber is fairly long, and it releases epinephrine or norepinephrine as the neurotransmitter. The parasympathetic system, seen in relaxation times, originates from the cranial and sacral nerves. The first neuron is really long and releases acetylcholine. The second neuron is really short, and it also releases acetylcholine as the neurotransmitter. I'm going to zoom in just a smidge, and I want you to make sure you get all of that information down about sympathetic and parasympathetic systems. And anytime you need to pause this and, make sure you get all the notes, please do that. So now, when we're looking at the sympathetic versus the parasympathetic system and where they arise, so over here is sympathetic, so I'll make a note of that. And over here is parasympathetic. So the sympathetic system you can see arises from the thoracic and lumbar regions of the spinal cord. And if we compare the first neuron or the preganglionic neuron, it's fairly short compared to the second one, which is fairly long. Okay. The first neurons here are going to release acetylcholine. The second one, once we get to the actual organs, those are releasing epinephrine or norepinephrine. And so those are going to cause that fight or flight response, increasing heart rate, increasing breathing, increasing blood pressure, causing your pupils to dilate, all of that stuff. I didn't mean to delete parasympathetic over here, so let me put it back. The parasympathetic system, we originate from the cranial and sacral nerves. So we see that here and here. This first neuron is really, really long. They don't even show the second one here because the second one is embedded within the organ itself. They show a second one here, but really it happens in the organ. Okay, so the first neuron is really, really long, and the second one is so short it's within the organ. Both of those release acetylcholine. Okay. And so these are going to stimulate the abdominal organs, decrease heart rate, decrease breathing, that kind of thing. So briefly, I'm just going to mention some of the neurotransmitters, just so you can have an idea. So neurotransmitters are chemicals released from neurons. The neurons have terminal buttons, and in the terminal buttons, you have vesicles. And vesicles, if you remember back to when you learned about the cell, are membranous structures that contain things. They can contain things that circulate around in the cell. They can contain things that have to be secreted, like in this case, neurotransmitter. So acetylcholine is going to be released for that preganglionic fiber for all of, whether it's sympathetic or parasympathetic, right? And they're going to be acetylcholine is going to be released by all of the neurons that are going to stimulate skeletal muscle fiber cells, or muscle fibers, or muscle cells. And so, two types of receptors that bind acetylcholine: nicotinic and muscarinic. So, nicotinic receptors affect our stimulatory and result in excitation. So, we see these at muscle cells and all ganglionic neurons. So, where we have that connection between one neuron and the next, acetylcholine is that neurotransmitter. Okay, and those are nicotinic receptors at that point. And then muscarinic are going to be inhibitory, and we see them at parasympathetic target organs, right? So then we slow down that activity. And then we have amines, example, epinephrine, norepinephrine, right? We talked about that with the sympathetic system. And then we have two types of receptors for epinephrine, norepinephrine, or amines: adrenergic receptors, alpha and beta. So alpha is stimulatory, and beta is inhibitory. Some examples of these, besides epinephrine and norepinephrine: dopamine, serotonin, histamine, other catecholamines, and dopamines. Then we also have amino acids as neurotransmitters within the central nervous system. Um, GABA, gamma aminobutyric acid, glycine, aspartate, glutamate. And I'm not going to get into a bunch of functions of these. There's a lot of information in your textbook on these. We just don't have time. The nervous system, you see that there are like 2 or 3 great big chapters on the nervous system. We're really just touching on this, so you just have some exposure. And then peptides are amino acid or protein based kinds of neurotransmitters, more protein. They're larger than just simple amino acids. And these have to do with pain or pleasure. Substance P, for example, induces pain. So when you eat spicy things, for example, you can eat something spicy and it's cold, right? So if you eat a cold salsa, but it's spicy, and you say it's hot, well, it's not hot in regards to temperature, right? But it's hot in regards to spice. What happens is that spice is causing the release of substance P, and that pain is what you're considering hot or spicy. Okay. Endorphins reduce pain. Other somatostatins stimulate intestinal activity. CCK, that was something you had before, stimulates intestinal activity. Okay, central nervous system. These are the things we talked about before. We'll put just a little bit of responsibility with each of these areas. So we have the cerebrum, the cerebellum, and the brainstem. The three major parts of the brain. So the cerebrum is important for speech, memory, thinking, emotions, consciousness, like being aware consciousness. The frontal lobe, in particular, gives us some control over skeletal muscle movement, also involved in speech and language. Our parietal lobe is associated with our ability to recognize pain, cold, touch, also some association with speech and taste. Temporal lobe, memory, hearing, and smell. Occipital lobe, totally dedicated to vision. The cerebellum, muscle coordination, motor skills, and muscle coordination. Cerebellum. Brainstem, consciousness, like being alive consciousness. All of our regulatory functions, like regulating blood pressure, regulating heart rate, regulating ventilatory rate. So our brainstem is responsible for that. So brainstem injuries can be deadly because that's the regulation of all those things that keep us alive. Okay. We already defined gyrus and sulci, or sulcus. The gyri or gyrus is a raised area. Gyrus is singular, gyri is plural. Those are the raised areas on the brain, and in between them we have those depressions or indented areas. Those are the sulci, which is plural. Sulcus is one. And then we have that central sulcus that divides the sensory from motor areas. So the precentral gyrus is motor, and the postcentral gyrus is central. And we looked at that on the anatomy video. So this is our cerebrum, cerebellum, brainstem, right? And we got our frontal lobe, parietal lobe, temporal lobe, occipital lobe. This is our central sulcus. This is our pre-central gyrus, which is motor, and our post-central gyrus, which is sensory. Okay, so that divides motor from sensory. Okay. The ventricles are those fluid-filled spaces. So here you can see you got the lateral ventricles within the cerebral hemispheres. And these ventricles have horns. We are going to name the different horns of the lateral ventricles. And then in between where the two hemispheres come together, we have the third ventricle here. And then the connection between the third and the fourth ventricle is the cerebral aqueduct. And then here's the fourth ventricle. And I'm using a blue pencil, so that's not going to be helpful. Let's change the color here. So we have our lateral ventricles in each hemisphere. Our third ventricle where the two hemispheres come together. Then we have our cerebral aqueduct that connects the third ventricle to the fourth ventricle. And then that leads to the central canal and the spinal cord. Let's zoom in. So here, inside of here, if I penetrate this membrane here, that's the septum pellucidum. And inside of there is a lateral ventricle. And then the space here is the third ventricle. This is the cerebral aqueduct. And the space here is the fourth ventricle. I want you to look at these ventricles, and you see that you have a network of blood vessels here, here, and here. Each one of these is referred to as a choroid plexus. And the choroid plexus produces cerebral spinal fluid. And so the cerebral spinal fluid is the fluid inside of the ventricles and inside of the central canal of the spinal cord. And so that fluid is important because the chemical changes in that fluid can actually trigger changes in various regulatory areas of the brain. Whether oxygen levels are high or low, CO2 levels are high or low, whether it's acidic or not, those kinds of changes in the cerebral spinal fluid can trigger certain regulatory areas in the brain. Go back to this one so we can look at those ventricles up close. Okay, we mentioned the blood-brain barrier earlier. Your brain is made up of a ton of neurons. Neurons don't undergo mitosis, so if we lose any, we don't get them back. So we need to keep that environment stable. We need to protect our neurons. We need to make sure they get adequate oxygen, adequate nutrients, and they are not easily damaged or exposed to things that might damage them. So we talked about the fact that capillaries are leaky, but the ones in the brain are the least leaky, right? We don't want things that get in our blood to easily have access to our brain. So we have the least leaky capillaries in the brain, And we have those astrocytes that wrap around the capillaries to make them even less leaky, to form what's referred to as a blood-brain barrier, to protect our brain from sudden, rapid changes. Then we have our spinal cord. We looked at the anatomy of this. The gray matter in the spinal cord is towards the inside. The white matter is towards the outside. The coverings over the brain and the spinal cord, those connective tissue coverings, are called the meninges. The dura mater is the outermost layer, and this covers the brain and the spinal cord. Dura mater means tough mother. It is tough, it's not stretchy. And that can be easily seen and cut away. And then the arachnoid matter is adhered to the surface of the brain and the spinal cord, but it can be teased away with tweezers, and it's kind of cottony or kind of looks like a spider web, thus arachnoid. And then the pia mater is closely adhered to the surface of the brain and the spinal cord. You cannot appreciate that with the naked eye, nor can you remove it. Now, let's talk about a reflex. A reflex is an involuntary response to a stimulus. An involuntary response to a stimulus we don't have control over. I don't have to think about it, it just happens. So I often ask my students, if somebody slaps you and you slap them back, is that a reflex? So we like to think that it is because you didn't think about it long, you just automatically did it, but it's not really a reflex. A reflex would be like if you touch something hot and you immediately pull your hand away without thinking about it, that's a reflex. So there are some more complex reflexes. We're going to look at a very simple reflex, something that just goes right to the spinal cord. You don't even have to think about it. A more complex reflex would be something like if you step on a tack, you got to pull your foot up, right? But in addition to that, you got to balance on the other foot. So that's a more complex reflex. But this is something really simple. So we have our incoming information here. So this is our, let's label some things here. We got this neuron, we got this neuron, and we got a pathway here. So we're going to label this pathway. We got a neuron here. We got something here. We got a name. And we're going to, let me get my pencil here. We're going to name this. We're going to name this. So let's name some of these things. So we have our motor neuron. Let me make sure I got a big enough text box. Sending information in via the afferent pathway. Here we have an association neuron or an interneuron. Okay. And at that interneuron, we're going to have analyzation and integration, or integration and analyzation. And then down here we have our sensory neuron. Oh, this is the motor neuron. Got to fix the top one. And this is the efferent pathway, or outgoing pathway. So up here is not motor neuron. So let me fix that. That's a sensory neuron. I don't know why I put that. That is a neuron that detects the change, and then that information has to come in. So let's fix that. Sensory neuron. So the sensory neuron is going to detect this incoming information, which is my afferent pathway, which is incoming sensory neuron. This information goes in right here. This is the dorsal root. This is the dorsal root ganglion. So let's label these things, and I'm going to change the color just so we don't have a bunch of stuff piled on top of each other, or at least if we do, you can see it. This is the dorsal root ganglion. Okay. And so then this is the dorsal root. And this is the ventral root. Okay. So my reflex has the incoming information coming in via the afferent pathway. This is a sensory neuron. It leads to my dorsal root ganglion and the dorsal root. That synapses in the gray matter of the spinal cord, sometimes with an interneuron, sometimes not. Sometimes it directly synapses with the motor neuron. Then we come out this ventral root. This is our efferent or efferent pathway, and this is a motor neuron sending information out. Okay. Then our cranial nerves, we looked at these on the base of the brain, and we numbered them and we put them in order. Now let's talk about what they're responsible for. Your textbook gives you so much more information. This is just a brief, quick view of what they're responsible for. This is all you need to know. So don't worry about all of that other information. Olfactory is 1 for smell, optic is 2 for vision, ocular motor is 3 for movement of eye muscles, trochlear is 4 also for movement of eye muscles, trigeminal is 5, sensory for face and motor for chewing. Abducens is 6, movement of eye muscles. Facial is 7, movement of facial muscles, and also stimulates the lacrimal and salivary glands. 8 is vestibular cochlear for balance and hearing. 9 is glossopharyngeal for swallowing and saliva production, also taste. 10 is vagus, and vagus sends motor fibers to the organs or viscera, right? So motor fibers to the organs or viscera. Viscera is another name for organs. And 11, accessory movement of some of the neck muscles, and 12, hypoglossal movement of the tongue. Now, there are little mnemonics for remembering this OOO, to touch and feel very good velvet. Which is the AH at the end, was something that I learned a long time ago. You have to just be careful once you remember the first letter of each one, OOO, to touch and feel very good velvet. So you got the OOO, TTAFVGVAH, what each one stands for, right? Because we got a couple of A's, we got a couple of T's, we got 3 O's, right? So you have to remember what those stand for and then what their responsibilities are, okay? And the last thing we looked at this for the anatomy are the plexi or the plexuses, and we have the cervical plexus, which originates high up in the cervical region. One of the most important nerves that comes off of there is the diaphragm, is innervated by the phrenic nerve. So the phrenic nerve goes all the way to the diaphragm, one on either side, and that diaphragm being innervated by the phrenic allows us to breathe. That's super important. Off the brachial plexus, we have the nerves that go to the arm, We got the axillary, the radial, the median, the ulnar nerves, probably musculocutaneous as well, that supply the arm. Then the lumbar plexus gives rise to the femoral and obturator nerves that innervate the anterior part of the thigh and leg. And then we got the sacral plexus that gives rise to the sciatic nerve that innervates the back of the thigh and leg. Okay. Make sure you jot down any questions that you have for me. And then our next notes will be um, Monday, the special senses anatomy, and Tuesday, the special senses lecture. But I want you to spend some time on this. I may post those earlier. We shall see. Okay? Jot down any questions you have. Make sure you take a look at the notes that are already posted and the models that are posted in e-learning.